Hi, my name is Mariana Nava, and I'm here to talk about the maguey Agave salmiana micropropagation. So, Agave salmiana, most commonly known as maguey, is a Mexican endemic plant which is mainly produced in the states of Hidalgo, Tlaxcala, Estado de Mexico, and Puebla. It has a medium to large size, and it has been used for textile production as well as pulque elaboration, and it has gastronomical or ornamental uses. It is a Mexican endemic plant that has the ability to adapt to stressful environmental conditions. It produces high quality meat. And also it is important to note that all its parts can be used, even its pest. By micropropagating these species, we'll be able to obtain populations with elite characteristics, as well as to obtain plants with antioxidant activity. It is also important to note the key role that Agave plays in the Sustainable Development Goal number 15, which is life on land. As maguey crops protect the land against erosion, it also captures and retains moisture that can be used by adjacent crops. And it also is host to animals and insects such as pollinators. It's important to note that the extracts obtained from agave sap and its leaves had been shown to obtain fructans and glycosides. And it is shown to have cytotoxic activity in these particular cell lines. Our objective is to design and implement a methodology for the in vitro micropropagation of agave salmiana in order to obtain good quality plants, to evaluate its potential cytotoxic activity, to treat HEC293 cells, ensuring that the findings will be beneficial to our training partner, WeClone. WeClone is the first commercial micropropagation laboratory here in Mexico. It is located in Querétaro, and it was co-founded by Dr. Ashutosh Sharma. Our hypothesis is that treatment with a specific combination of hormones, AAA and BIP, will positively influence shoot formation and efficient multiplication of agave salmiana plants in a micropropagation system. For the methodology, we're going to be using different media preparations, as we're going to have different methodologies. This is due to the fact that we had a lot of contamination and oxidation, and we needed to change this cleaning process and the media itself to have better results. The first preparation of media contains only MS, the hormones at the different concentrations shown here in the slide, and also agar, and the pH was adjusted to 5.7. For the second preparation, we decided to add activated charcoal to help with the oxidation problems, and two different antifungicals, Captan and Velate. However, the antifungicals resulted in lowering the pH to 2.1, which meant that the media did not solidify, so it was useless. So for the third preparation, we decided to remove the antifungicals. For the fourth methodology itself, we decided to use a combination of the third and first preparation, as we first of all cultivated the explant on the media with activated charcoal, and after a few days, we changed it to the media without it. For the cleaning process itself, on the first methodology, we had quite a general cleaning process, and it was shown not to be enough to remove the contamination and the oxidation issues. So for the next cleaning process, that was to methodology number 3, we decided to add longer periods of time in the washes. And we also added 1 hour wash with antifungicals, 2 different antifungicals, Benlate at 2% and Captan at 2% also, and a 1 hour wash with microdin at 2%. It's important to note that for the second methodology, we did not have a cleaning process as the media did not solidify, so we were not able to use it. And for the fourth methodology, which is this is the final methodology that we propose, we decided to add an antioxidant bath to combat the oxidant problems. So for this methodology, we first selected the mother plant, which was done in this case by our training partner. We removed the leaves and the roots. We had a superficial wash with Twin 20 and then a 30-minute wash with Twin 20 at 2%. We then performed the extraction of the merismatic explants. We had a 1 hour wash with antifungicals, 2% Velate and 2% Captan, as well as 1 hour wash with microdin at 2%. We then had 35 minutes wash with 40% chlorine. And finally, an antioxidant bath with citric acid and ascorbic acid at a concentration of 150 milligrams per liter. We then removed the damaged tissue and we placed the explants on the media. For the cytotoxicity assay, we're going to use the HEC293 cells, which are human embryonic kidney cells. For the extract preparation, we're going to wash the leaves. We're going to dry them at 40 degrees Celsius for 8 days. We grounded them, and when we mixed 10 milliliters of methanol with 1 gram of powder. We placed this on the sonicator and we centrifuge it, and we finally filter it to sterilize it. And this extract was stored at 4 degrees Celsius. For the cytotoxicity assay, we're going to be using 4 different concentrations, as well as a positive control and a negative control. And we're going to read the absorbance at 517 nanometers after 24 hours and after 48 hours. For the results, we can see that in the first methodology, we had a lot of contamination. We had bacteria and fungi, as well as oxidation. For the second methodology, unfortunately, the media did not solidify, as by adding the fungicides, we lowered the pH to 2.1, which meant that it was not able to solidify. On the third methodology, we had a positive result, 1 healthy explant that showed shoot formation, but we also had some fungi and a lot of oxidation. This was resolved by adding the antioxidant bath, and we had a lot of positive results, a lot of healthy explants. However, these explants were only healthy for around 2 days. After these 2 days, they started to have a lot of oxidation, as we can see here on the left. So after 4 days, we decided to subculture these explants to a media with only MS and the hormones. And we also decided to subcultivate the healthy explant of the third methodology. After 6 days of this subculture, we can see that all of the explants from the fourth methodology resulted in necrosis, but the explant from the third methodology still survived. So we can see that mainly on the early stages of the protocol development, we had a lot of bacterial contamination, and this contamination was present in all of the explants. Therefore, we can conclude that it was probably due to endogenous bacteria. In Agave salmiana, this bacteria is Bacillus, Enterobacter, and Klebsiella. The fungi problem, the fungi was present in only a handful of explants, which means that it's most likely due to poor aseptic technique. The oxidation, which is the browning of the plant tissue, is a frequent problem in vitro culture, and it is due to the damaged tissues. The damaged tissues can be a result from an aggressive disinfection process, as well as a result from the cuts made to the explant itself. And these damaged tissues lead to the production of the phenolic compounds, which are absorbed by the medium, and they inhibit growth and lead to tissue death. For the cytotoxicity assay, we can see that the percentages of viability after 24 hours and after 48 hours were always above 70%. This means that the concentrations used were not cytotoxic. Therefore, if we want to do further research, it is important to note that a different extra preparation is needed, as well as higher concentrations and more time of exposure. For the statistical analysis, we wanted to first of all test the difference between the mean of the number of shoots formation per explant in the different treatments. But sadly, this was not done due to the fact that we had no growth, as we had a lot of complications with the cleaning process itself. But ideally, if we had obtained this data, our response variable would have been the number of shoots grown per treatment. We will have one factor, which is the BIP concentration, and 4 different levels, which leaves us with 4 different treatments. And for the cytotoxicity assay, we wanted to test the difference between the mean of the absorbance value read at 517 nanometers for the different concentrations used. But this was also not possible due to the fact that we had not enough repetitions. So it was not possible to do an ANOVA analysis. In conclusion, we can see that we were unable to test the effectiveness of the treatments due to contamination and oxidation problems, but a cleaning method was discovered to successfully address the bacterial and fungal contamination in the explants. However, we will need to do further research in order to solve the source of the oxidation itself. Also, for the cytotoxicity, we will have to do further research and further tests with different concentrations of the extract, or we should use another plant source to fully understand the cytotoxicity of Agave salmiana. And also, it is needed to make more replicates and to continue researching and testing to obtain significant data that will allow us to make a statistical analysis. That will be all. Thank you very much for your attention.